Our research question has been if male homosexual behavior is a feminized one, and in this movie we will try to explain and answer this question by portraying different points of view. Our initial viewpoint was that male homosexual behavior is not a feminized one. The basis for this was that we suspected that this was based on a common stereotype aimed towards homosexual individuals, and that there are probably bigger intragroup differences between homosexual and heterosexual men. We also believe that if there are behavioral differences, these were more likely to be due to environmental and social factors. We also thought that it was important to think about what femininity is and what it actually means. We thought of it as a behavior that is typically displayed by women and that is connected to gender roles. Traits that are often used when describing women could be, for example, more emotional, empathetic, sensitive, and more aware of their outer appearances compared to men. The subject of human sexuality has been a hot topic for research for many years now. As our initial viewpoint is only based on our own assumptions and experiences, it's important to see if there are any studies answering the question. We will here describe a few problems with the hypothesis and present some biological approaches to the topic.
One of the most common biological theories explaining sexual differentiation is the prenatal hormone theory. The hypothesis is that prenatal androgens play a crucial role in the development of sexual differentiation in both the central nervous system and for behavior. According to the theory, the principle states that increased prenatal androgens cause a masculinized behavior and states that decreased prenatal androgens feminize behavior. So how can this be connected to sexual orientation? There has been some evidence that prenatal testosterone could play a role in generating male-typical sexual orientation. The theory has further support looking at neuroanatomy, where some differences have been found between hetero and homosexual individuals. For example, a difference between the sizes of the third interstitial nucleus of the anterior hypothalamus was found, which is smaller in homosexual men compared to heterosexual. The size of INAH3 is similar between homosexual men and women. Amongst these studies, the effects of prenatal androgens and different hormone sensitivities have been studied and seen as a possible clue to these differences. However, more research needs to be put into this area, and most likely, hormones are not the only explanation. Several animal studies have also shown that perinatal sex steroids have a large effect on the mate choice within several species. One study of a sheep breeding facility showed that 16% of the male sheep did not mate with female sheep. Out of the 16%, 10% of the male sheep showed a homosexual behavior as they mated with other male sheep. 6% did not mate at all. In the amygdala, a level of estrogen binding was found which resembles that of females and which is much lesser than that of heterosexual male sheep. Furthermore, lower testosterone levels were found in the homosexual rams. In general, there are behavioral differences found between men and women. They differ in levels of aggression and childhood play behavior. Usually, men express higher level of aggression compared to women. There also seems to be gender differences between the ways children play. Girls have been shown to show preference for toys such as dolls, dressing up items, and makeup, while boys prefer toys such as cars and weapons. Boys also tend to prefer more rough games such as wrestling and playful aggression. So what's the case with gender-type behavior and sexual orientation? In one study, it was found that there was a significant relationship between gender non-conforming behavior at the age of 3.5 to almost 4 and homosexuality at the age of 15. The gender non-conforming behavior includes, for example, differences in toy preference. In another study, researchers found a connection between gender-type behavior of men and sexual orientation 5 and 6 years later. They measured the level of femininity in adult men using a questionnaire and found that this was a predictor for future homosexuality. Another weak point of our initial viewpoint is that there were neural differences regarding the brain structure found between homosexual and heterosexual individuals. One study conducted that the brain of heterosexual males and homosexual females show rightward cerebral asymmetry. Furthermore, the brains of homosexual males and heterosexual females show a symmetry of volumes of the cerebral hemispheres. In addition to this, sex atypical connections were found in the amygdala of homosexual individuals. While the connections were more widespread from the left amygdala in homosexual males and heterosexual females, it was the opposite side in homosexual females and heterosexual males. Moreover, there were differences about which parts of the brain displayed the connections. Again, it was the same for homosexual males and heterosexual females, as well as for homosexual females and heterosexual males. According to Savik and Lindström, the results indicate that they are neurobiological entities. Taken together, these findings do not answer the initial question properly, but still might favor the assumption that homosexual males show a feminine behavior. Reviewing the material, we can see that there is research pointing in a more biological direction as we initially assumed. There is no doubt that homosexual and heterosexual males differ regarding their femininity. It was found that homosexual men's childhood play habits were more feminine in their execution, that also as adults, homosexual males are behaving in a more feminine way, and that the brains of homosexual males are structurally similar to the ones of heterosexual females. These studies provide a foundation indicating that homosexual males do exhibit a more feminized behavior, which could be caused by both biological and environmental factors. A biological factor could be a difference in prenatal estrogen production. Still, it has to be emphasized that future research should put more focus on these factors, as there are a lot of different biological explanations, some even very contradictory. But also our new position has some limitations. Nelson and Kriegsfeld state in their book An Introduction to Behavioral Endocrinology that different hormone concentrations causing homosexuality is a common but largely unsubstantiated hypothesis. According to them, many studies have failed to find any consistent evidence on this. They declare that the findings are not reliable. Gonadotropin response to estrogen treatment is not a stable predictor for homosexuality in men. In addition to this, they affirm that tight controlling in human studies is difficult, which questions the validity of the studies. Besides, if different hormone concentrations lead to homosexual males being more feminine, it is hard to explain how it is possible that quite a lot of homosexual males do not look or behave feminine at all. One example for this is the bear community. Its members are usually homosexual or bisexual men who are larger, obese, and with a lot of hair, spreading a picture of pure masculinity. In addition to this, having the findings of the sheep study in mind, it is important to ask if the difference between 6% and 10% is significant and if you can transfer the conclusion to human sexuality. A general issue looking at the studies presented is also that it's much harder to test these hypotheses in human subjects, since it would be unethical to regulate, for example, hormonal levels in pregnant women. Furthermore, many of these studies are built upon small sample sizes and retrospective measurements, which opens up questions about validity and reliability.